Proteomics is the large-scale study of proteins. Proteins are vital parts of living organisms with many functions. Proteomics is the large-scale study of proteins, as we discussed. What is proteins, polypeptides, peptides, and study of proteins. The proteome is the entire set of proteins that is produced or modified by an organism or system. So then the question arises, what is proteome? Proteome is the complement protein found in single cell in a particular environment. It is complete collection of proteins encoded by genome of an organism. Proteomics is the study of composition, structure, function, and interaction of the proteins directing the activities of each living cell. In actual, the non-amino acid part or non-protein part is known as prosthetic group that helps to work protein properly or help in the function of a protein, like heme molecule, zinc, and iron, and so many other molecules like zinc finger proteins if not be functional without zinc ion. So these are considered as prosthetic group or cofactor. Heme group is also discussed here. A prosthetic group consisting of a protoporphyrin ring and a central iron atom, that is Fe. Cofactor is a non-protein chemical compound or metallic ion that is required for an enzyme's activity as a catalyst, a substance that increases the rate of a chemical reaction. What is proteolysis? Proteolysis is the breakdown of proteins. Degradation, the protein degraded into different components, into amino acids for different purposes, for different functions. So protein degradation is the degradation of proteins into amino acids for any functions or for other purposes. The protein degradation is very important. If we have so many proteins in a cell or in an organism, we don't need that. We just need a specific number or specific quantity of proteins. Same about the protein turnover. The organism always carry the protein turnover. That is important. The number of proteins important for the specific functions. That is known as protein turnover. When older proteins are broken down into the body, they must be replaced. This concept is known as protein turnover. Two main mechanisms of protein degradation, in which one belongs to the lysosome, that is engulfed by, that is also known as autophagy process, with the fuse of autosomal with the lysosome and formation of autolysosomes. And another one is the cytoplasmic complexes. As you remember that the birds are generating and just engulf the material to degrade. Sometimes there are some proteins to degrade. There are different types of proteins, and every protein has different rate of turnover and degradation. Depends on the nature of the protein and the demand of the cell and the organism during the process of translation on ribosomes. Okay, protogenic amino acids are amino acids that are incorporated biosynthetically into proteins during translation. The word protogenic means protein creating. The amino acids help to make proteins. There are how many amino acids we have? We have 20 amino acids, and sometimes people call it 22 amino acids, but usually we have 20 amino acids. So these helps the binding of protein and the creating of protein. Non-protogenic amino acids are those that are not naturally encoded or found in the genetic code of any organism, despite the use of only 22 amino acids by the translational machinery to assemble proteins. And it's a very good information here that over 140 amino acids are known to occur naturally in proteins, but we only study about 20 amino acids usually, and thousands more may occur in nature or be synthesized in the laboratory. I also mentioned some links here for your knowledge. Protein is the basic unit for every function in our body is performed by proteins with one or another way. A linear chain of amino acid residues is known as polypeptide, and a protein contains at least one long polypeptide. Short polypeptide contains less than 20-30 residues. All the proteins have interactional studies with proteins, DNA, carbohydrates, lipids, and RNA, and all the functions performed in our body by different molecules and different macromolecules depends on proteins and interactions of proteins and communications of proteins. So abundance in cell, now we are on abundance in cell. So abundance in cell, it has been estimated that every cell size in bacteria contains 2 million proteins per cell. So this is the information about how many cells and how many proteins in how many species are here. So we just discuss it in a very brief manner. Like bacteria contains almost 2 million of proteins. Now we are on biosensors, like we already discussed that different peptides and amino acids and polypeptides we can synthesize in lab with the help of experiment. And chemical synthesis, short proteins can also be synthesized chemically by a family of methods known as peptide synthesis, which rely on organic synthesis techniques such as chemical ligation to produce peptides in high yield. After that, we have post-translational modification. The protein works after the post-translational modification when we have different types of attachment of different chemicals, different compounds, and also the deletion of different chemicals and compounds. Glycosylation is the covalent attachment of oligosaccharides and addition of glycosyl group at asparagine and serine and threonine, and it helps for the in the folding of protein, correct folding of protein, conformational changes of the protein, distribution of protein in the cell and organism, and most importantly, stability and activity of the proteins. Attachment of nitrogen to the asparagine or arginine side chains, so it will be N-linked glycans. The attachment of phosphate group at the serine amino acid is known as phosphoglycans, and then we have C-linked glycans, that it is a very rare form, and sugar is attached to the carbon at tryptophan side group. Ubiquitin group, small protein attached for the destruction of protein, or you can say engulfment of protein or degradation of protein. It is ubiquitination. Then we have S-nitrosylation, that is nitrosyl group attached to protein. Nitric oxide compound is also involved in this one. It is a chemical messenger, react with free cysteine to form S-nitrothiols.